Hi there. Apologies for being a few minutes late this morning. We're having a few technical difficulties. Really happy that you're back to join us for the third of the BSGCT's translational seminar series in collaboration with Catapult today. Today, we're going to be talking about the non-clinical development of cell and gene therapies, and we're going to be joined by Keith McLuckie and Matthew Smart, and they will take it from here. Thanks very much, guys. Over to you, Keith.
Thank you, Alex. I trust that everyone can hear me. So yeah, thanks for being invited again. You may, if you're on the last call, the last webinar, you may have heard me talking about the strategic and regulatory considerations for ATMPs. So now today I'm going to talk to you really about the non-clinical aspects, and later on Matthew is going to talk to you about the production development. Okay, so let's start with the first slide. Okay, how can we best assess the safety and biodistribution of my product? That's what you all realistically want to know. So from an academic point of view, this may be fairly daunting, but realistically, it's fairly straightforward. You just need to follow this pipeline here. So at the top, we have, essentially the ATMP roadmap to adoption. So this starts with preclinical studies, clinical development, marketing authorization, and then ultimately you get your therapy into patients. So from a non-clinical aspect point of view, we're really talking about the early aspects here. So you have your academic research, that's all about proof of concept and efficacy. Then you move into the safety, so that's all about toxicology, which is potentially the daunting part, and then that gets you to your phase 1 clinical trial, your first in human clinical trial. And the non-clinical space is really a continuum throughout this space. So you may have in vitro studies and in vivo studies, and that brings you to the clinical phase. So that is the forward translation. So this is supporting clinical development. You will be looking at your mechanism of action, exposure, target engagement, safety, pharmacology, and efficacy. And that gets you to your first in human clinical trial. And this is a space where obviously you're in humans for the first time and there may be issues. And so if there are any toxicity issues or any mode of action issues, you can then use reverse translation, is where you really go back into in vitro models, in vivo models, and you really try and tease out the toxicology or the mode of action, or really what you're trying to do in the clinic. So the reverse translation, this is clinical data feedback and various different investigations to support the clinical phases thereon. From an ATMP, largely speaking, your phase 1 clinical trial is going to be a chronic and a fairly large affair from a duration point of view. So you may not need necessarily the standard phase 2 and phase 3 preconceptions that you would do for small molecule or biologics. And that is really what you have to discuss with the regulators of the MHRA, for instance, in the UK. So really here you are it's essentially a feedback loop, and you are using in vitro and in vivo work to help you understand the clinical space. So if we move to the next one. So to get into the clinic, you need a certain amount of non-clinical data. So you need to demonstrate a proof of concept in a relevant model. So this may be in vitro or in vivo proof of concept studies, in vitro or in vivo efficacy studies. So for instance, if we take a CAR T as an example, you may have a cancer xenograft model where you have cell lines that are expressing your CAR target, and therefore you can use that for your efficacy modeling. And then we need to think about the support for the use of the administration route and application procedure. So I mentioned this in last week's talk, but essentially you need to think about what you're going to give the patients in the clinic, and you need to make sure your dose route is suitable in your preclinical space. So is the therapy going to be administered by IV injection? Is it intracerebral injection, or is it direct into a tumor? If you have some sort of novel route, i.e. you've got some sort of medical device, this is where you need to think about how that really can be used in your early on studies. You then need to think about the support of the selection of a safe and biologically effective starting dose for your clinical phases. So after efficacy and proof of concept studies, you will have dose range finding studies and then your pivotal GLP safety studies. So these should really be designed to support a sensible and safe clinical dose. And this also needs to demonstrate your mode of action. And obviously from an ATMP point of view, it's not really about putting as much as you can into the subject or the model. It's a sensible system where you don't obviously have too much from a price point of view, but you do need efficacy and prove your mode of action. And then finally, you really want to look into potential appropriate safety data for clinical monitoring. So there may be some biomarkers of disease halting that you can take through to the clinical phase. There may be pharmacodynamic endpoints that you can use for monitoring the clinical phase. For instance, if you've got a gene therapy, you have a transgene, so you can look for upregulation of the transgene or expression of the transgene. If we're talking about some sort of CRISPR situation where you can realistically look at knockdown of your target, and that can be justified from your preclinical space to your clinical space. So, as I mentioned before, the clinical, non-clinical, and CMC aspects all come together to help you along this work stream towards the clinic. So, how much should you know about your product? So, really, a slight change of mindset is required in the translational space. So, from an academic point of view, it may be all about, fancy graphics to put in a Nature paper, which everyone is, essentially pushing for. But from a regulatory point of view, it's not about the graphics, it's about the standard modeling. And realistically, it's a yes, no. Is this toxic in this model? Is this safe in this patient? So it's a different sort of mindset. So the academics really need to understand that you're you're moving over into a completely different mind frame. And regulatory bodies are there to keep patients safe, and they're not there to obstruct your innovation. So ultimately, you need to tell your development story. So you start off with your proof of concept studies. So these can be in vitro or in vivo, and you follow the mode of action using the appropriate model, whatever that is. So, essentially the gold standard from an academic point of view. You then move into efficacy. So this can be in vitro or in vivo, and you prove your mode of action treats the chosen indication, also in an appropriate model. And then finally, you go through the safety, biodistribution, and tumorigenicity modeling. This can potentially be in vivo, but CAR T is CAR Ts are a separate subject anyway. So you prove your mode of action has no untoward toxicity in normal, healthy, appropriate chosen animals. Obviously, across the board, there may be exceptions, but that is generally what you are really needing to look into. So you need to look at on-target toxicity, off-target toxicity. If we're talking about some sort of cancer indication, you really want to look at on-tumor and off-tumor issues. So these three phases here, you could realistically use 3 different models or the same one. You really have to pick what is appropriate for your product and the indication, and every ATMP is essentially bespoke. So what sort of studies do you need? So in vitro or in vivo pharmacodynamic proof of concept studies, as I said before. So looking into transgene expression or CRISPR knockdown or stem cell engraftment. Bioactivity and pharmacology profile of the product. You may want to look at the level of gene expression if required. So you want to essentially look at how much expression you get and how much expression you will need to cure that disease. So how much expression do you need in your model? How much expression do you need in your preclinical studies, your safety studies, and therefore how does that relate to your clinical phase and how much expression would you need there? And there's also the extent of pharmacological correction. So if we're talking about some sort of CRISPR mechanism where you're knocking down a certain gene, how much of knockdown is appropriate? How much do you need? Is it 10%? Is it 20%? So you really need to know, how much is enough, and that can essentially come from your proof of concept, your efficacy, and your safety to really get into the clinical phase. So the toxicology studies are for the assessment of safety and tolerability. And then you can use biodistribution studies to look at the persistence, the clearance, and mobilization of your cell therapy or your gene therapy. So for gene therapy, you have your vector and your transgene. So there's quite a lot known about AAVs. So realistically, that is not going to be the driving force of any toxicity you're going to see. From an AAV point of view, yes, it may go to the liver, there may be immunogenicity, but it's really the transgene that is going to push any issues. So that is what you really need to concentrate on. From a cell therapy point of view, ideally you would want engraftment. If you're talking about, say, a stem cell, you want engraftment at the site of introduction, and then the stem cells will do their job. So if you have engraftment at a distal site, there's the potential of tumorigenicity depending on the mechanism, but also that means that there may be a reduced efficacy at your target site. So there's two issues to this that you need to realistically think about. So toxicology and biodistribution studies can often be combined into one study. So this realistically is going to be a 6-month study, and you want to get as much data out of this as you can. So think about that before you go to this stage, because you cannot really come back and repeat studies in this space. Another aspect you need to think about is safety pharmacology. So central nervous system, cardiovascular, and respiratory endpoints. So a therapy is not necessarily going to be a success in the clinic if it induces heart arrhythmia or some equivalent severe response. Obviously, this really depends on the indication and the target, and if you can mitigate for this, but safety pharmacology is an important way to mitigate risk for your patients. And then finally, immunogenicity. So obviously, from a point of view of stem cells, there's potentially an issue with immunogenicity. So you can run cytokine response assays to look at that. And from a gene therapy point of view, you will have potentially antibodies to your AAV, which is potentially always going to be an issue. So these are sort of things you need to think about. So let's talk about test item considerations. So you've heard me talk about GLP and GMP before. So GLP is good laboratory practice, and that is really how you regulate running the safety studies. GMP is good manufacturing process, and that is realistically how you regulate the production of your therapeutic agent. So the material used in safety studies should be well characterized. That is the main take-home message here. It needs to be well characterized. It should be comparable, if not identical, to that you're proposing to use in the clinical trials. So the GMP grade material with a nice certificate of analysis and good batch release status is the gold standard. But you can mitigate for any differences. You really have to justify to the MHRA or whatever regulator you're going to as to why your product is suitable for the studies you're going to do, and why you're taking this decision. So impurities from the GMP production batch compared to an R&D batch, realistically may be an issue on safety. So purification processes should remove them rather than attempt to qualify them in a safety testing program. So for instance, if you've got an R&D batch that is 90% pure, and then you move to your GMP batch that is 99% pure, if you run your study with the 90% R&D R&D product, you have 9% that you do not know what's happening. So it could give you an increase in your pharmacology. It could give you an increase in efficacy. But bearing in mind that from a gene therapy point of view, your safety aspects are really pushed by the adverse pharmacology. So that's why you should really know that the product you're going to put into your patients is safe from this point of view, because it's similar to what you put into the preclinical models. So that is the end of my bit on the non-clinical part. So Matthew will now talk to you about the production considerations. So over to you, Matthew, and I will sign off.
Thanks, Keith. Good morning, everyone. So just moving on from Keith's section, I'm going to talk as Keith highlighted. I'm going to talk a little bit more about how you get to that process and how you actually make the product that you're going to need to get to the business end and hopefully treat the patients. We're all excited to help. And I think really what I wanted to do is just set the scene and pose a few ideas and sort of hints and tips for things that can really help you as you progress through your development and move through clinical trials and ultimately all the way to commercial authorization and widespread usage of your product. And we find that a lot of developers are kind of very focused, which particularly in the ATMP space makes a lot of sense. Very focused on just getting that initial clinical data, and I think that's reasonable. And then worrying about potentially dealing with some of these more complicated issues around manufacturing later on. But actually, I think if you spend a bit of time thinking about it early, particularly in that discovery phase, when you're developing these new therapies, you're making choices around, what serotype to use, what capsids to use, what cell type to make. Actually, can really save you a lot of time and issues later on, and actually issues that do, ultimately cause a lot of promising therapies to fail before they reach the patients that they're intended to help. So I'm just going to cover a few of those things and kind of talk from my experience in terms of the types of pitfalls I think maybe early developers, certainly those in an academic space that perhaps are undergoing that mindset shift that Keith spoke to when moving into the translational phase with their technology. So, as I say, I think the first element is to try and have a good discussion with your team, with your subject matter experts, as early as possible. Engage help where you're not clear and on what the path looks like. Certainly, that's what the Catapult's here to support you with. And then also, I think one of the key messages to get across is you need to protect your technology very, very early as possible. So that's not necessarily talking about patenting and things like that, but I think you can seek advice from your tech transfer officers or your IP lawyers and really get an understanding of what you should and shouldn't disclose. And that's really, really critical. We regularly see really promising ideas and technologies that just really struggle to have a path forward, because ultimately you're going to need some private investment to be able to move these therapies all the way through clinical trials into the patient. It's just the reality is just too expensive. You're not going to necessarily get there on grants alone. And without that commercial proposition, you're going to be unable to do that. And that's not to say you can't publish. We're really keen, I think, for everyone to publish and get their ideas out in the public domain, but just doing so in a way that is structured so it protects you and your technology is going to be really important if you're actually going to realize the potential of those technologies to help patients. So that would be my number one thing. And now there are many others, and I've covered a few on this slide, but I think it's often good to think about what you're doing from a more industrial standpoint while you're developing things. So very basic stuff is, while you're developing it, are you keeping good records of your information? Are you keeping good data? Are you making sure that you can understand what material that data was generated from? So, for example, if you're going to do an early, proof of concept animal study that shows efficacy or safety with your product, have you also characterized that product before it went into the animal, rather than doing maybe a batch and running some PCRs or some flow cytometry or some other analytical technique, and then finishing that experiment and then making another batch and putting that into the animal. Being able to connect those two data sets is really critical, really important, and it's going to really help you when you have to look back at your historical data to answer questions that pop up during your preclinical and ultimately your clinical development. So it's about making sure that you've got that traceability, you've got those records. It's not just stored, in your favorite postdoc's head or your PhD student's head. It's somewhere where it's easily accessible. It can be transferred and put in front of people who can then obviously help you, who maybe sit outside your organization. Other things might include things like, how reproducible is my process? So reproducibility is a huge issue in ATMP manufacturing. There's a lot of green-fingered elements. We often have certain people that are very skilled and they become our kind of primary manufacturers while we're doing the preclinical development. But actually, as you move towards the clinic and ultimately commercial manufacturing, to be able to serve the patient population you're trying to help, you're going to need something that is a little bit more simple, ideally, something that is robust. It's going to make the same product over and over again. I liken it to, it's almost trying to grow the same plant twice. If you're doing perhaps a pluripotent stem cell differentiation, for example, and you need that plant to be able to do the same things, grow the same fruit every single time. So if you can structure things so they're robust and repeatable, you understand where those potential failures when perhaps you transfer it to a new PhD student in the lab, or you've shared your protocol with another group and they've had issues getting it up and running. I think having that confidence around the reproducibility of your manufacturing process is really critical. And again, that's something that we regularly support therapy developers with. Slightly kind of adjacent to that, I think, is how predictable your process might be, and what mechanisms do you employ to be able to predict what's going to happen. Now, manufacturing a lot of these ATMPs is very, very labor intensive, very expensive, and usually quite long. And that's always going to then feed into the complexity of the risk of failure, and also the cost of these therapies cost a lot of money. We need to make them cost effective for healthcare services to be able to access them, and ultimately to get a widespread uptake from those patient populations. So if you can use a tool, use an analytical system to be able to tell you, well, actually on day 2, if I don't see this marker, for example, I know on day 35 I'm not going to get what I need. You can fail early and fail cheaply, and then restart the process and move forward without having lost too much resource, too much time, and ultimately, you're able to then, in a manufacturing sense, still hopefully meet the deadlines you need to be able to help your patients. And again, it might be completely critical if you're manufacturing something where, perhaps an immunotherapy, a CAR-T product where, your patient is actually in a terminally ill situation if they don't receive that therapy as soon as possible. So these things, do really matter, not just from an economic perspective, but also from a patient perspective. It also will help you and speed up your development if you can shorten certain outputs. So if you can, optimize your starting material, for example, your cells that you're using, or maybe your vector manufacturing, it'll help you to do much more development more quickly, more screens, more optimization, if you've got reliable in-process indicators about how the end product's going to look like. So that speed is another key driver in these early development phases. The investment often comes with some milestones, usually first in human is a key one for early development. And you probably won't be given as much time as you'd like to get there. So anything like that, that when you kind of go into that translational phase that you can bring on board is really key. I think another key aspect that is often overlooked and it's often kind of disregarded is how do you know when the process is going badly? And actually, what does bad look like? Is there various shades of grey, or is it black and white? What aspects of that process cause things to go off the rails? And actually, that data and following that data through and putting those bad cells into your proof of concept models is really, really valuable as you move through your development, because what it allows you to do then is have a baseline, have a reference for, you know, firstly, how sensitive is your manufacturing process to being able to make a product that is efficacious and safe. So it gives you an idea of that design space that you're working with, but also, you know, it allows you to develop those analytical tools to be able to nudge things back on track aren't going so well. And ultimately, you know, moving through the various phases of development, we really want to move away from the labor intensive, very hands-on type of manufacturing process and bring in more automation and more decision making that is not necessarily made through experience, but it's actually made through, you know, data. So that allows you to then eventually bring in technologies to do automated feedback and change the process based on live information that it's measuring as it progresses. So having the good and bad and the light and the dark there with your product is really, really useful as you go through your development. There are other things that a lot of people are kind of already live to, but things like are your reagents and your products used to manufacture your cells or your vector animal product free? Are they xeno free? A word of warning, I think, and from experience on some of these things, some of the, you know, the CTS grade or the GMP grade inverted commas materials you might find aren't animal product free. They may have trace elements from their manufacturing. You really need to dig down into that and do a raw materials assessment as you're moving forward towards the clinic, because you will find some surprises. And again, there's lots of places, including the Catapult, that have kind of been through that process over and over, and they sort of know where a lot of those skeletons are hidden. So they can advise you on what things might cause problems. But ultimately, it's not necessarily a complete barrier. The classic being BSA or, you know, serum in your processing. It won't stop you necessarily moving forward for clinical use, but it will probably cause you a lot of hassle. And you'll need to demonstrate that those products are safe. You may need to acquire at great expense large lots of those types of products to be able to show a consistent manufacture across what you need for a batch, for example. So it will cause issues. So if you find you can remove them as you're developing your processes, then that can be really advantageous and streamline things later on. A couple of other things moving on to my kind of next slides are standardization, really. And again, we see this quite commonly, is that perhaps actually no one's actually really ever written the protocol down. And actually no one's ever written it down, and actually different people are kind of doing different things at key steps, and maybe they're eyeballing something, they're making decisions, they maybe do slightly different things that they've kind of adapted based on their experience. And actually that can make it quite difficult, I think, again, around that data integrity question that we talked about, in terms of, well, actually, what process was used to make the cells that went into your proof of concept animal model, for example? And actually, do we find that, you know, different experiments that we're using to justify the potential of our therapy are actually made using slightly different manufacturing processes? And can we rely on the data and being able to compare those different data when we're taking that forward to regulators? So I think that standardization can also bring consistency, but it also locks down the idea of, okay, this is how I'm doing this batch of cells or this batch of gene therapy product, and and then I've got a really clear idea of what went into my my endpoint assays or my animal studies, and then by all means, your process will change. It's inevitable, will change from there. But at least you can do that in a controlled and iterative way that can bring confidence and bring confidence in the eyes of a regulator, to be able to justify, delivering this to human patients. So once you kind of had a good think about that, well, you're developing it, you're making those decisions. Maybe you're having a look at the GMP reagents that you're selecting. You're not assuming that the GMP reagents are going to work the same as the research grade reagents. Again, another absolute classic thing that we find all of a sudden, the cells start behaving differently because actually the manufacturing for those reagents is slightly different. Once you've kind of considered a lot of those things, I think it's also good to be thinking about, what does actually my product look like? And I'm talking about a detailed description of what your product looks like, not necessarily just, okay, it's this gene I'm going to replace, or it's this cell. It's actually, no, it's this cell, it's this number of cells, and it's for this intended indication, and it's going to be treated at this type of stage in the disease, and it's also going to be delivered using this device or this needle, and it's going to be delivered in this excipient, for example. It's in saline, or it's actually going to go straight from thaw, so it's in a cryopreservant. And what are the implications of that for the patient? And actually having that end goal in mind is absolutely critical, because it can actually really heavily influence what your manufacturing process might look like. And I'll give some examples, but ultimately what that means is you really need that clinical input early on when you're thinking of translating your therapy. And actually, it's more common than you might expect. People necessarily on our side of the fence, on the scientific side, we don't always do this. And actually, it can have quite large impact, I think, on how you would progress the translation. You may find that it throws up issues that take a lot of optimization and take a lot of time and resource to get right. So the earlier you identify those, you're in a good place. You've got the runway to be able to invest and develop those and overcome those barriers before it becomes a terminal problem for your development pipeline. So I've touched on some there, but I think it's a good idea to think about what you need at various stages of your clinical development, your translation, in terms of what does your batch size look like? How many doses is a reasonable amount of dose? That's going to be informed by the numbers of patients available to you. It's going to be informed by the cost of goods and the remuneration that you can receive from healthcare providers. Perhaps it's going to be available if you're treating a rare disease, like a lot of the gene therapies that are on the market. Are you going to have the stability if you're only going to get a patient a couple of times a year? Are you going to make one big batch and store it, or are you going to make smaller batches? That can, again, look very differently. So it's good to have that in mind. A classic issue is the cryopreservation versus fresh. Now, it's hard to know in a kind of general sense which is the better option. For a lot of rare diseases, a lot of immunotherapy products, for example, the autologous therapies, fresh seems to work fine. You can get the supply chains in place to be able to turn that around and deliver those to patients. Obviously, it's quite complex and it has its issues. It also often requires quite a heavy QC burden. So the release of each product has to be released usually in an autologous sense for each product. Whereas if you can do a larger allogeneic batch that treats lots of different patients, you can reduce the QC burden, because you can kind of do all the testing on a larger batch once, and then you can infer that the rest of the doses are going to be okay. So your cost of goods comes down. You get that economies of scale. But that said, if you've got that larger batch, unless you're going to have a very, very large queue around the back of your manufacturing facility, the likelihood is you're going to need to deliver that to a point of care, and you're not going to necessarily be able to have all those patients for each batch. Maybe if you're treating thousands of patients on the same day or in the same week, so you're probably going to need to store that, and you're probably going to have to cryopreserve it. So that can often cause massive problems in terms of, you know, the phenotype of your cells, the amount of cells that you recover post-thaw. Are you going to need to be able to recover the cells and deliver them directly in the clinic? That probably means you need to start talking with a technology provider who can develop a device that's going to facilitate that for you. Perhaps, you know, you have 50% losses when you thaw, so you need to actually figure that into your calculations in terms of your batch size that you need to manufacture to be able to meet your dose. So all these things are very interlinked, and ultimately they're going to be really, really important for getting you where you need to be. Some of the more kind of unseen issues that I've seen involve things around stability. You know, is there a hold time? Does it have to sit in a, you know, does it have to go up in a lift? Does it have to sit in the clinic for 12 hours because the procedure is very long? Some of the logistical concerns that come with manufacturing a clean room is you ordinarily wouldn't be allowed to have the liquid nitrogen in the clean room. So if you need to freeze something, snap freeze something immediately, you know, it might be, I've heard as high as 45 minutes to get that out of the clean room facility, transfer it through all the airlocks to the warehouse where the liquid nitrogen store is. So you need to make sure that your product is going to be stable during that period, in case anything gets held up, or, you know, someone falls ill and can't deliver it in time, or whatever it might be. Or find a device again that can freeze, that is able to be used in the clean room, and can mitigate some of those issues. So I just really wanted to give you all a flavour of, it feels very complex. There's a lot of different interconnected considerations to make, but I think in principle, if you take that endpoint in mind, you think about, okay, what does this actually look like? How many cells am I going to want to deliver? How am I going to deliver them? You know, is the needle going to pop off my cells when it's being injected? Then you can get a really solid idea of, okay, so what needs to sit behind that to get me to that endpoint where, you know, the cells or the vector is, you know, is going into the patient and hopefully doing the brilliant job that we expect it to do. And if you do that, you've got a much higher chance of being successful and hopefully treating those patients and making them well. So I think that's everything from my side. But thank you for listening, and obviously happy to take any questions.
Thank you very much, Matt and Keith. That was really great overview of what is a very complex area. A few questions. First of all, going back towards the beginning of the session. So Keith, you mentioned about combining your phase 1/2 and how that with CGT products, you don't necessarily require that traditional structure in quite the same way. Can you expand a little bit on this and when you think it might be appropriate to confirm? I appreciate that this is someone for the regulators as well, but any examples you can think of?
Yeah, well, essentially, the old school way of thought from a small molecule point of view is you would have a small study, maybe into healthy individuals, but that's not the case from an ATMP. Realistically, you're going to go into patients and people that are really, you know, potentially sick in the first place. So from that point of view, you can think about phase 1 to combine sort of approaches, and really carry on with your cohort that you have in your phase 1 study can then potentially carry on down the line into your next studies. So really, because of the therapies we're talking about, either, you know, for CAR T cell, it would be a cancer indication. For some sort of rare illness, there may be issues with that. So I guess, as Matt said, if you talk, you know, talk to clinicians, talk to KOLs about what is in the space for your indication in the clinical phase, they can really help you understand, yes, we can sort of push this really accelerate this from the clinical point of view. But you don't have to always think that, oh, it's going to be phase 1, phase 2, phase 3, just as the old school 20 or 30 years ago from a small molecule point of view. Essentially, everything to do with ATMPs is scientifically discussable. So you justify as to why you want to do this. And as long as all the way through your development, you are proving that the safety is mitigated or it is as best as it can be, and you are doing the best for the patients, and they are essentially in on this from a rare disease or a cancer indication point of view. So yeah, talk to clinicians early on, as Matt said, and you can really work out. And you may think, oh, this is easier than I really anticipated from those phases.
Brilliant, thank you. And a question for Matt as well that we have. You mentioned that it's equally important to investigate what a bad process looks like as to what your outcomes are. What do you think a bad process looks like, and what are the problems that you might encounter?
Well, ultimately, I think as developers and subject matter experts in these different areas, I think you're best placed to tell me or people like me who are trying to support you what a bad process looks like. So I think the best way to think about it is if you think about what the quality profile of your product should be for it to be ultimately safe and efficacious. So anything that you think is going to cause issues or inconsistency in meeting that criteria that you set out, then that potentially is an area for focus when you're looking to try and tighten up and develop your process. So bad can be anything from not meeting the yield that you need for your batch. It can be something that impacts the phenotype of your product. So perhaps your cells don't grow in the way they should, or they don't differentiate in the way they should, or your vector doesn't contain the correct amount of cargo or the correct cargo. So anything like that means that you're not getting what you want at the end ultimately. And as I said, these are big processes. There's likely lots of different causative effects that could lead to that. That those are interesting. And I think what's good about keeping a record of things that have gone wrong or look bad while you're developing things is actually they're going to be way more representative of what it's likely to be an issue that will arise. If it has arisen, it's likely to arise in the future as a potential failure point for your manufacturing. So it's a good sort of foundation for you to then target your process development to understand, okay, how do we avoid that happening, and how do we monitor that it's not happening now? And I think that gives you a really solid foundation for you to be able to progress.
Thank you. And to both of you as well, a question about preclinical and non-clinical models. You mentioned that it's important to select your pharmacodynamic endpoints that you're going to be monitoring in the clinic, but presumably this requires validation in your preclinical models as well. How do you select the best possible model? And I imagine the academics are probably going to be well placed to help guide what this would be. Are there any situations where there are no appropriate models?
Yeah, essentially the time that is the sort of bane of our existence when we are sort of trying to help people with this. So if you're talking about a gene therapy, you think about what your target is. You want your target to be in the model that you're using. So you look at homology, you do the various different searches to prove that your gene is within this species, and that's great. From a point of view of animals used, realistically, everyone would prefer to use rodents rather than higher animals. But if there's no pharmacology in rodent, you are not going to help from a safety aspect point of view, because as I alluded to before, for gene therapy, it is your target homology, it is the pharmacology of that product within the animal that is really pushing the toxicity. So also, as I said before, the efficacy models and the proof of concept models, they do not have to match the safety models. From a point of view of this pharmacology pushing the toxicity, as long as your safety model is really looking at the pharmacology and you pick that model with justification, that's what you really need to do. So you might have a knock-in or knockout model or a humanized model for proof of concept or efficacy, and then you move to a normal wild type model or complete different species for your safety. So that is tricky. And when it comes to something like a CAR T cell, you know, the ones that are out on the market for CD19, that was sort of very difficult to find a model where realistically you can do safety studies. So there may well be a situation where you can justify to the regulators that there's no pharmacology in any given species apart from emu or something, which you're not allowed to use. So therefore you say, right, I'm going to go with my in vitro data. I'm going to mitigate for going into the clinical phase and then use my clinical pharmacology to really justify moving forward. But obviously you need to discuss that well, you need to justify the science, and that is realistically a case for these, you know, fairly well, sometimes difficult products to work with.
Wonderful, thanks. So there really is a mechanism still, though, for taking forwards products which may have difficult models or no model in the non-clinical stage, as long as you have a suitable in vitro package and you can justify it well. Okay, and one last question as well that we've got. There's obviously a lot of different technical expertise that's required to do this. Is Catapult able to help facilitate conversations with relevant parties who are going to be experts in each of these areas, from model selection through to process development?
Absolutely.
Yes. Go on.
Go for it.
You go first, Matt.
Yeah, no, absolutely. We essentially cover the full gamut of translational activities. So we cover everything from those early kind of looking for investment. So we have a commercialization of research team where we're looking to support early developers or spin outs with putting together a reasonable commercial opportunity that makes it more investable. We help to understand what the investors are looking for and help you to kind of address those gaps that you might have. We also do early process analytical development, so taking a process from an early stage and developing it to a point where it could then be used in a clinical setting. We provide, and I'll kind of leave Keith to kind of talk to this, but we also provide all the kind of regulatory, non-clinical, health economic support as well to help you understand, what models you're going to need, what reimbursement price or model you should be looking towards for a particular indication, whether or not it's going to be something that healthcare providers can viably purchase and give to patients. It's really important. And then also we've got teams around skills and things like that to help you to help upskill the industry. So there's a good skills base for people to serve the growing industry. Large scale manufacturing. So we have a Stevenage and a Braintree site, and also a new development lab in Edinburgh. So we've got a kind of through line. So you kind of do your early process development, and then you can move through into manufacturing clinical material, and ultimately potentially beyond into commercial manufacturing. So we really do try and do the comprehensive support for everyone. And I would highly encourage you, if you are unsure or slightly mixed by some of these conversations and some of the lingo, then please just knock on the door and we'll be more than happy to shepherd you in the right direction depending on your needs. That's exactly what we're here to do.
I guess Matt introduced me to do carry on talking, but he really explained everything to you fairly well then. But just come to us if you've got any issues with any of the talk. Well, this week's talk or last week's talk, just get in touch and we can sort of chat and help you through any implications that you, or any worries that you really have with your ATMP development. So
Wonderful.
Nice to chat.
Thank you very much, guys. So yes, please do reach out to Catapult if you have any further questions, or come to BSGCT. We're happy to help connect you to the relevant people as well. I think that is a wonderful way to wrap up this seminar. And thank you very much both. We'll be digging down further into some of the issues that came up today, especially spinouts and IP in subsequent seminars. But our next translational research seminar will be looking at a case study of people who have gone through this process and how that looked for them. We'll reach out by email and on social media to tell you the exact time and date for that. Thank you very much both, and hope to speak to you again soon.
Our pleasure. Thanks for listening, everyone.
Thank you.
Thank
Goodbye.
you all.